Hello students, welcome to EPG Pathshala. My name is Dr. Priyanka and I teach Anthropology at the Department of Anthropology, Dr. Hari Singh Gaur University, Sagar, MP. Today I am going to teach about the module on definition and scope of population genetics from the paper Human Population Genetics. So let's see what we are going to learn from this module. We will be knowing about the origin and growth of theories in evolution. We will also be knowing about the various evolutionary forces that shape genetic variation. By the end of this module, you will be able to know the definition and scope of population genetics, its history, and some of the very famous mathematical models of population genetics. So let's begin. Modern humans evolved in Africa about 200,000 years ago, and they migrated across different regions of the world. While traveling through diverse conditions, they underwent modifications both culturally as well as biologically to adapt with the environmental pressure for their successful survival. These modifications left traces of variation in the human genetic structure. Thus, studying the genetic structure of human populations can help us understanding these modifications as well as the reason behind these modifications. Genetic evidence are given by patterns in variation in complete or a specific region of human genome. That means if you want to see the genetic evidence of any evolutionary force, you need to screen either a region of a DNA or you can scan the complete genome. It is useful as genetic evidence bear imprints of the earliest evolutionary events. We also call these genetic imprints as genetic signatures of the evolution. However, screening genetic variation without knowledge of other factors such as geography, environmental factors, ethnohistoric information can lead to an incomplete understanding of this diversity. That means, along with genetic information, we also need information from other backgrounds such as geography, the ethnohistory of a population, the environment in which it has lived or have passed during the course of migration, and etc. An anthropological approach is therefore very useful in gaining insight on genetic diversity by considering a range of factors to apportion the quantity of variation observed. Dobzhansky rightly said that nothing in biology makes sense except in the light of evolution. So in case if we are observing any biological phenomena, we need to understand them in terms of evolution. Evolutionary forces subject human population to change in biological traits. These traits are inherited, that means they are passed from one generation to the next generation. Studying these biological differences among human population in an evolutionary framework comes under the purview of biological anthropology in general and population genetics in particular. Population genetics deals with the assessment of genetic difference underlying biological traits. We estimate allele and genotype frequencies and predict the way they would change or remain constant over a period of a time. Evolution basically means genetic change. So now let's apply in terms of allele or genotype frequency. If during the course of time there is a change in an allele or genotype frequency, we can say that evolution has occurred. However, if they remain constant, we say that the population has not undergone any evolution. The alternation in frequency are used in mathematical models to comprehend the interplay between different microevolutionary forces. It is not always that only the single microevolutionary forces is acting upon the population. There can be many microevolutionary forces. So it is always the joint effect of all these microevolutionary forces which brings alternation in the frequencies. The patterns of genetic variations are used to test hypotheses on demographic events that have occurred during the history of modern humans. So, Before we proceed to understand the broad definition of population genetics and what are its various mathematical models, we need to understand about the evolutionary forces. And before we understand the evolutionary forces, we need to understand that how they were developed. The first noteworthy attempt to explain the process of evolution was given by Lamarck. Lamarck theory is popularly known as theory of inheritance of acquired characteristics. Lamarck's theory, also called as Lamarckism, is primarily consists of two main principles. One is a principle of use and disuse of organ, and the other is the inheritance of acquired characteristics. Lamarck reasoned in his theory that organisms adjust to their environment, and while doing so, modifications occurs in them. That means there is a change in the environment. This change leads to the modification in the traits. The resulting modified traits are then passed on to the offspring, making them better adapted. Although Lamarck correctly emphasized on the presence of animal-environment interaction, he was wrong about the evolutionary changes occurring in an organism lifetime. Understand this in terms of some examples. Lamarck says that there is a change in the environment. This environment sometimes leads to the overuse of certain organs, whereas it also leads to the not use of certain organs at all. He gave the example of giraffe's long neck. When environment changed, there was a pressure on giraffe to stretch its neck so that the giraffe can eat the leaves on the long trees. So as a result of overuse of a neck, the neck of the giraffe gets elongated. Similarly, let's understand the disuse or not non-use of certain organs. We all know about appendix. Appendix is that portion of the intestine which is not used at all. So therefore, now appendix exists as a rudimentary organ in man. These organs, which are either very much used or not used, basically make the organism better adapted to that particular environmental setup. And these characteristics are then passed on to the next generation. Therefore, the principle of inheritance of acquired characteristics comes into the play. So we need to remember these two main principles while we are understanding about the Lamarck. Darwin's theory of natural selection have been influenced by the work of many, including geologist Charles Lyell, who endorsed the idea that a complete understanding of the history of Earth requires knowledge of the ancient geological changes. Likewise, Darwin's theory of evolution was propelled by the work of naturalists, geologists, and other population theorists. A large body of evidence from biogeography, embryology, and anatomy, however, helped him to formulate his theory on evolution. His work on the origin of species have laid down on the three main important observations. The first observation was that all organisms have the ability of explosive population growth that can use up all the food resources available. This idea was borrowed from an essay on principle of population given by Malthus. A second observation was that the population growth was, however, stable. Based on these two observations, he deduced that populations do not attain their maximum potential to reproduce, and only few individuals who successfully reach a reproductive age are selected for. Darwin observed that minute differences existed even among individuals in the same population. He implied that some of these variations might be favorable as against the others and support perpetuation of the organism. Darwin received similar inputs from the field biologist Alfred Russel Wallace on the mechanism of evolution. In essence, species could be perceived as entities in constant interaction with their environment and responding to changes therein. In this interaction, natural selection filters out the traits that did not favor the organism's reproductive success. One of the important feature of natural selection is the successful transmission of adaptive trait to the offsprings. However, Darwin's theory of evolution by natural selection fell short of plausible explanation for this hereditary transmission. He understood that natural selection operated in a population due to the variation, but Darwin could not explain the origin and mechanism of the transmission of the same. Meanwhile, Gregor Johann Mendel gave the idea of hereditary transmission of traits of inheritance through observation made during his breeding experiment on pea plant. Based on these observations, he proposed the particulate nature of hereditary transmission. He postulated in organism hereditary characteristics are controlled by a pair of factors which we today known as allele. He further said that in the event of existence of two different factors for a characteristics, the one which is dominant is expressed while the other which is recessive is not expressed. The another law which Mendel gave is a law of segregation, which was proposed to explain the dynamics of factor during gamete formation. Mendel proclaimed that the pair factor undergo random segregation with each gamete receiving a factor with equal probabilities for each other. The last of the Mendel's law is called as law of independent assortment, according to which segregating pair of factors assort independently of one another. Mendelian law of genetics laid the foundation for genetic research by giving a roadmap for the identification of phenotype as genetic features by following three patterns of inheritance. However, unfortunately, Mendel's work was neglected initially and rediscovered by De Vries, Correns, Teschermack independently in 1900 through their breeding experiments on other species. So, we have three main theories of evolution. One is a theory of acquired characteristics given by Lamarck. Then, the theory of natural selection developed by Charles Darwin by the influence of many other scholars and work of others. And finally, we have a theory of Mendelian inheritance which was given by Gregor Johann Mendel and which was rediscovered by De Vries, Correns and Teschermack. Population genetics is a branch of biology that provides deepest understanding of how evolutionary changes occur. Population genetics can be broadly defined as a branch of genetics concerned with the hereditary makeup of population in a mathematical way. It involves the study of genes and genotype frequencies and then predicting the way these frequencies will change or remain constant under the combined influence of various factors over time within and between the populations. Population geneticists usually focus their attention on Mendelian population, which is defined as a group of interbreeding, sexually reproducing individuals that share a common gene pool. Population genetic study is relevant today as its theory, along with the technological inputs from the other branches such as molecular genetics and bioinformatics, is contributing immensely to document the allele frequencies at polymorphic loci at a large scale. This information is used to infer past demography and the effect of evolutionary forces operating on human populations in effective ways. Now let's see what are the various evolutionary forces that can shape the genetic variation. As we know, evolution in a broader aspect can be defined as encompassing change over time. In the context of evolution, existence of genetic variation becomes immensely important as they form the substrate for evolution. The key forces that shape the pattern of genetic variation are mutation, recombination, genetic drift, natural selection, associative mating, and migration. The first one, which we are going to understand, is recombination. Recombination is the exchange of segments of homologous chromosomes during meiosis. It leads to an increase in a genetic diversity due to the formation of new allelic combination. Alleles in closely spaced loci do not undergo random segregation during meiosis as a result of infrequent recombination. This is why evolutionary forces acting on one locus might affect the adjoining loci. In this context, two events have been observed: one, hitchhiking, and the other, selective sweep. Hitchhiking results when positive selection at a locus leads to rise in the frequency of another allele, and selective sweep occurs due to reduced genetic diversity at loci linked to a recently fixed allele. The next evolutionary process which we are going to discuss is mutation. Mutation is a random change in the actual genetic code, including change in a single DNA base, insertion or deletion of a DNA sequence, and other rearrangement of DNA sequences. Differing by the number of bases affected, mechanism of mutation, and region of localization, these variants exist as SNPs, insertion deletions, STRs, VNTRs, and CNVs, inversions, and translocations. Mutation is a primary source of variation and has a significant impact in the process of evolution. A mutant that are found in less than 1% of population is called as rare mutation, whereas if its frequencies rises above 1%, we call it as polymorphism. The next evolutionary force in the line is genetic drift. Genetic drift is a stochastic process in which a subset of a population undergoes mating that results in the allele frequency change in the next generation. At the level of an entire population, this means that each generation may not have the exact same set of allele frequencies as the previous generation. Drift increases with the reduction in the size of the population and causes changes in the allele and genotype frequencies over time. Either of the allele is eliminated or is fixed as equilibrium is reached, but it is difficult to predict the identity of this allele due to randomness of the nature of this process. The other evolutionary process is natural selection. Natural selection, as we all know, is a Charles Darwin's contribution to evolutionary theory, referring to differential survival and reproduction. It occurs when there is a difference in fitness for different genotypes, such that there is a change in allele frequency over time. The change in color of peppered moth in England is an example of natural selection. Natural selection acts upon phenotypic variation to bring about the difference in reproductive abilities of individuals with different genotypes. The rate of evolution by natural selection is proportional to the genetic variation in fitness. The viability, mate selection, fertility, and fecundity together constitute the fitness of a genotype, and its relative fitness in comparison to other genotypes is given by the selection coefficient. When a mutation leads to less fit genotype, it is subjected to negative or purifying selection, while one which increases fitness undergoes positive or diversifying selection. The another evolutionary factor is gene flow or migration. Migration is an inter-region movement of people and causes gene flow when migrants contribute to the next generation in the new location. Gene flow balances out genetic differentiation and lessen allele frequency differences between the populations. Several population geneticists have proposed models to explain the gene frequencies caused by the gene flow. One such model is island model proposed by Wright. It is the simplest model that shows a meta-population divides into smaller population islands of equal size which exchange genes at the same rate per generation. Likewise, there are other models such as stepping stone model and isolation by distance models, etc. Let us have a brief description of the historical background of population genetics. The initial development of the field of population genetics took place well outside the field of anthropology. The idea proposed by Darwin and Mendel remained incompatible for a very long period of time. Later, the ideas were synthesized to explain the genetic transmission of natural selection for evolution mathematically by three people, namely R. A. Fisher, J. B. S. Heldon, and Civil Wright. Much of the core of population genetics can be traced back to these three men in the early part of 20th century. Over time, their mathematical formulations were combined with observation from laboratory experiments, field studies, and the fossil records to develop what is often referred to as synthetic theory of evolution. Let us learn about their contribution individually. R. A. Fisher. R. A. Fisher was an English statistician, evolutionary biologist, mathematician, geneticist. He developed some of the concepts such as balanced polymorphism, heterozygote advantage, ANOVA, and maximum likelihood estimation of linkage. He showed how correlation between relatives can be explained by cumulative effect of large number of Mendelian factors, each having a small effect. Civil Wright was an American geneticist. He pioneered the now standard tool of population genetics, inbreeding coefficient, and F statistics. He gave the concept of civil right effect. He also propagated the interplay of genetic drift with other evolutionary forces. For this, he devised the method of path analysis and used path coefficient to describe the outcome of interbreeding. Assortative mating and selection came his seminal paper about the title Evolution in Mendelian Population, in which he established the maladaptive changes preceding higher levels of adaptation. J. B. S. Heldon. Heldon was a British naturalized Indian scientist well known for his work in physiology, genetics, and evolutionary biology. His research was important for his stress upon the quantification of the rates of change of a population's characteristics, and he introduced quantitative approach for estimating human linkage map using a maximum likelihood method and mutation rate. Though three of them contributed immensely towards population genetics, there were, however, academic differences between the three. The most conspicuous difference was the three's opinions on natural selection being the most important factor in shaping of genetic structure of a population. Fisher and Heldon supported natural selection as a main driving force for evolution, whereas Wright believed in the chance factor and migration contributing towards genetic variation. Beside these three scholars, the foundation for population genetics was laid in 1908 when G. H. Hardy and Weinberg independently published their work, which later became famous as Hardy-Weinberg equilibrium. The Hardy-Wimbuck principle enabled estimation of genotype frequencies of the succeeding generation given the allele frequencies in the parental generation of a population. Hardy-Wimbuck proportion for the two alleles can be depicted graphically. The horizontal axis shows the two allele frequencies P and Q, and the vertical axis shows the expected genotype frequencies. Each line shows one of the three possible genotypes. The graph shows that given the allele frequencies, expected genotype frequencies can be calculated and vice versa. The increased molecular geneticist abilities to generate large amount of polymorphic data posed a huge problem. The models proposed by Fisher, Heldon, Wright could not account for the enormity of polymorphic data, the explanation for which was given by Kimura. Kimura proposed neutral theory of molecular evolution. This theory explains that the change in allele frequencies are a result of random and neutral process of genetic drift and not the different types of selection. The theory is regarded as a null model of molecular evolution. Polymorphisms will ultimately be fixed or eliminated when they are dictated by genetic drift. The deleterious alleles might undergo negative selection, but positive selection and balancing selection rarely occurs. The theory also explained the constant rate of nucleotide and amino acid substitution in lineages over time. This realization formed the basis of the molecular clock hypothesis that is utilized to time evolutionary events. Extensive data on DNA variation provided another major impetus to the field of molecular population genetics with the introduction of retrospective models to learn about this history. This huge amount of polymorphic data was utilized to study the past events like events of inbreeding, gene flow, or natural selection. The coalescent theory was proposed to reconstruct the ancestral state of population starting from its current state. This theory was given by Kingman. The theory was based on the idea that all genes in a population are derived from a common ancestral gene, which is called the most recent common ancestor, and the time to coalescence as time to most recent common ancestor. Thus, in this session, we have reviewed the contribution of various scholars towards the field of population genetics. So let's summarize the entire session of this module. Population genetics can be known as a study of heredity in organism in a mathematical way. Population genetics usually focus their attention on the Mendelian population. By the term Mendelian population, we mean a group of interbreeding, sexually reproducing individuals that share a common gene pool. Gene pool, as we know, is defined as a sum total of all the genes and their alleles which are present in a population. Population genetics is highly mathematical and make use of allele and genotype frequencies. The various mathematical models are used to understand the mechanism of evolution. Evolution in a broader aspects can be defined as a change over time. In this context, existence of genetic variations becomes immensely important as they form the substrate for evolution. In population genetics, we study the change in gene or genotype frequencies, which is the indication of evolution. In population genetics, we study this change in genotype and gene frequencies, which is the indication of evolution. The change in the gene frequencies can occur due to the operation of various mechanisms which we call as evolutionary forces. We have also understood in this module that the key evolutionary forces that can shape the pattern of genetic variation are mutation assortative mating, recombination, process like genetic drift, natural selection, and migration, or which we also know as gene flow. There are various theories regarding these evolutionary forces. The theories of evolution were given by the works of many scholars, such as Lamarck, Charles Darwin, Charles Lyell, Gregor Mendel. However, the two main important theories are given by the Darwin and the Mendel. These ideas given by Darwin and Mendel were entirely valid, yet they remain incompatible for a very long period of a time. Then ideas were synthesized to explain the genetic transmission of natural selection for evolution based on mathematical models. These mathematical models were given by Fisher, Halden, and Wright. These are the pioneers of the population genetics. Much of the core of population genetics can be traced back to these three men in the early part of the 20th century. However, the work of Hardy-Wimbuck laid the foundation of population genetics. The Hardy-Wimbuck principle enabled estimation of genotype frequencies of the succeeding generation given the allele frequencies in the parental generation of a population. The law, however, have certain assumptions, such as mating should be random, the population should be large in size, there should be no operation of evolutionary forces such as mutation, selection, drift, or migration. If nothing happened, then it means no evolution has changed place because you will not see any change in the allele frequencies. But if allele frequency is found to be changed from one generation to another, that means evolution has been operated on the given population. The field of population genetics is full of such mathematical models with the help of which we can have a better understanding of evolution.